Hi.
Hi. How are you?
Good. How are you?
I'm wonderful.
Good.
It's life.
So let me tell you a couple things before we get started. We're going to almost finish histology today. We'll finish it on Thursday and then start bone on Thursday, which is good because you guys are doing bone, but you'll be doing bone lab for 2 weeks. So, a couple things I want to tell you. First off, in addition to this, which is all the open lab times, please keep your eye right down here because starting, I hope this week, they're going to get back to me soon, at least at some point in the near future. There will be lab aid-led study groups meeting weekly on Redwood and Jordan campus. And that's part of the projects that I require them to do. And you are invited to go to those. And it's basically just that you have someone who was successful with the class and has had some training in teaching there with you to answer questions. And you can go through lecture and lab material with them, not just lab material. So I'll post them there as soon as I get them finalized. And then in addition, the STEM Center on Redwood campus is also doing topic-specific workshops. Like I think next week there's one on Axial Skeleton in which like somebody just like sits with you with skulls and goes through them. And that's taught by one of the lab instructors who used to be a lab aide, Andy. So I'll just post those there when they're there. And hopefully, have you had some time to rummage around on the site and find all the stuff that was there? There's a bunch of stuff there. It really is. Go ahead.
I have a question. How do we gain the password for the restricted area?
Your lab instructor will give it to you.
Okay.
As soon as you learn everything you're supposed to learn about proper care and respect of cadavers. Then you can get in there. So they should probably give it to you this week because there are pictures of the skeletons in there. In addition to they're protected for the body donor program. I had to ask special permission to photograph them at all because generally it's not allowed. And then he said that I could photograph them and label them. The advantage there is that those are our cadavers in the pictures and not just an idealized one that's in the textbook. But I had to keep them behind a password wall, and you guys had to have some education about cadavers before you could get access to them. So that's it. And the ones that don't let you print very easily, I mean, you can always get around it, but if they don't let you print, it's because the body donor program didn't want them printed because that one was, for instance, too much like the actual donor, right, or something. So there's some that are easily printable and there's some that aren't. Primarily the easily printable ones are the ones that you can practice and label on. You can write, do it over and over again. Okay, so that's that. Is there anything else, any other concerns that you guys have? Okay, so you guys are starting on Axial Skeleton this week. This is the week that it's going to start feeling intense. So just a couple of little things about that. It's not just you. Everybody feels overwhelmed. It always happens. It happened to me. I literally thought, what the living hell with how much crap that I had to learn. But you can totally do it because people who are dumber than you have done it. Okay? I've had some students in the past that probably didn't know the right way to screw in a light bulb and passed anatomy. So you guys can do it too. It's just going to take some repetition and some practice. And so I'll just tell you a couple things. First of all, sometimes it's hard to convince myself to memorize a bunch of things when they seem arbitrary. So let me tell you a little bit about the stuff that you are being asked to memorize. You are being asked to memorize the mastoid process of the temporal bone because I'm going to attach the sternocleidomastoid to it. You're being asked to learn all those damn holes because cranial nerves are going to go through some of them, and the carotid canal has the internal carotid artery that supplies 75% of the blood supply of the brain going through it. So the things that you are going to be asked to learn now seem arbitrary. I promise you I took as much of the arbitrary crap out as I could, right? Eventually they will reinforce each other and you're going to sort of see them repeating, right? So you're not going to just learn all the holes of the skull and never see them again. You're going to learn all the holes of the skull, and then when you learn the cranial nerves, you have to figure out which hole it goes through, right? So you're just going to have to trust me for a minute. It's like reading a Stephen King novel. Have you guys ever read a Stephen King novel? It starts out with 5 parallel stories, and you're like, I'm going to trust you, but what the hell do these people have to do with one another? And then they all come together, and shit goes down, right? Same thing in anatomy is what's going to happen. So yeah, it does. I know it seems like a hell of a lot. It will come together quite a bit better than it initially seems like it's going to. Okay, so where we were last time is we were doing all of the connective tissues. All of the connective tissues are derived from this tissue. What is this one? What is it? What? Common origin. Where would I find that? Would I find that in Charlie right now? No, it's embryonic, right? This is embryonic connective tissue, the precursor of the connective tissues. These are the adult connective tissue categories, right? And we were working on proper, which is the squishy one. This is the relatively solid one, and this is the fluid one, right? Okay, so we're going to finish the propers today. We did irregular and adipose last time. The two categories of connective tissue proper are loose versus dense. Somebody tell me in your words, what does this categorization refer to? Loose versus dense fibers. Yeah, the density of the fibers in the matrix, meaning how many fibers versus round substance, right, do you see when you look at it? So we were doing the loose connective tissues. We've got irregular, adipose, and reticular of the loose connective tissues. So let's just look at them. Now, adipose doesn't look like it's supposed to. We'll get to that in just a second. This one, does it look like a loose connective tissue versus, for instance, this one? Yes. Yeah, right. So you can tell the difference between a loose and a dense. And what about this one? Loose or dense? That's loose. Yeah, that's loose, right? Now, contrast that one, if it doesn't make sense with you, to you, with this one. Does that one look loose or dense? Dense. Do you see? Right? If it's really, really difficult to find any place to label ground substance, yes? That's a good indicator that you're probably not looking at a loose connective tissue. Yes? Okay. So the loose ones that we looked at last time were irregular, and so this is the picture from your textbook, right? And that one's easy. Can you label 1, 2, and 3 for me? What is 1? Elastic fibers. What is 2? Collagen fibers. What is 3? Fibroblasts. What is 4? Ground substance. Good, right? Get confident with that one. What if it's a slide you haven't seen before and is stained slightly different than you expected? Can you still tell me what this one is? Yeah. This is irregular, right? Somebody go through for me your criteria in your head that go on for identifying irregular.
Is there ground substance?
Is there visible ground substance? What about what else? What else makes you know? The fibers, right? Different kinds of fibers going multiple different directions. That's probably what I would identify, right? Okay, so, and then this one's a bit closer up than you than you're used to, but it still is irregular, right? It's still irregular. Okay, and then that one's also irregular. That's a pretty slide. I like that one. And then we were doing adipose last time, and we can all agree that adipose doesn't look like a typical loose connective tissue. Does anybody remember how I explained to you how you go from something looking like this to something looking like this? How does that happen? Why can I not see appreciable amounts of collagen, elastic fibers, and fibroblasts in here? Jocelyn, were you going to answer? Go ahead. Oh, I thought you said. Go ahead. Yeah, the cells move in and they kind of squash everything between them. So even though this doesn't look like a typical loose connective tissue, and this isn't a lumen, is it? What is it? That's a lipid vacuole, right? And it's not empty. It's not the absence of something. It's the presence of something that doesn't stain because it's lipid soluble. It doesn't stain with protein staining. So this is a lipid vacuole. What's the whole cell called? Adipocyte. And no, I won't make you label it on here, but I want you to understand that because this is a connective tissue, it also has fibers, although they're difficult to find in there. It has elastic fibers and collagen fibers. It even has some reticular fibers, but you're sure as hell not going to have to label them, right? So what is this whole cell called right here? Adipocyte. And it's really vascular tissue, so you can, a lot of times, if you look closely enough, you can see blood vessels actually running through adipose. So adipose is really vascular. And have you ever seen liposuction go on? Yeah, seen videos of liposuction. You can see how bloody that tissue actually is. It's really, really vascular tissue because as you grow more adipocytes and they go through cell division, they actually cause what we call angiogenesis, which is they cause more blood supply to develop into them to sort of feed them. Okay, so last thing is, where do you find adipose? You find it wherever loose connective tissue is, but primarily in the subcutaneous layer under the skin, right? That's where, so you can tell the difference between loose irregular connective tissue. This is loose irregular connective tissue. Most of us don't have very much adipose on our forearms at all, right? But almost all the women in here could pinch our thigh, right? And where the loose irregular connective tissue is primarily adipose. Because, of course, estrogen causes increase in fat tissue in certain locations in women in different locations than men. Right? That makes sense? Okay. And so the other places that you have this, it's less about energy storage and more about sort of padding. So for instance, it's around your kidneys, on the surface of your heart. It also is in your joint cavities, especially joints that are under a lot of pressure. So inside the joint cavity as padding between the bones, you have a little bit of adipose. And when you lose weight, you don't usually lose that sort of organ and joint fat first because you actually need that. You lose subcutaneous fat long before you lose organ and joint fat. When you start losing organ and joint fat, it's usually because you're kind of wasting away. Like if anorexia or chronic disease or something, you'll start losing that as well. Okay. And then the last of our loose connective tissues is reticular loose connective tissue. And reticular loose connective tissue, get a good look at it. So what it has is all these dark fibers, but they're not straight. They're kind of, they look kind of like a hairnet to me, right? Because it's primarily what kind of fibers that you're seeing there.
Reticular.
Yeah, it's reticular fibers, right? It's the only slide that you can dependably label reticular fibers in. And then the nuclei that you see of the cells, and the cells are either, most of them are actually leukocytes, but you can call them fibroblasts if you want to, but the cells are nice and round. I don't usually make you label the cells on this one, I'll tell you, because people don't understand spleens yet. But can you tell that this is also reticular connective tissue?
Yes.
Do you see the hairnet with the nice round cells in it, right? And how about this one? This one's further away, so you might have to stretch your mind a little bit. Yeah, you can still tell, right, that it is hairnet with nice round cells in it. Okay, so what does this tissue do? Where do you find it? This is, so you got a squishy organ like the spleen or a squishy organ like the liver, okay? The internal framework that helps hold that loose sort of arrangement is made of this kind of scaffolding. So think of it as the internal framework inside the spleen that has the spleen sort of hold this loose shape, okay? But the difference between the spleen's internal framework and the liver's internal framework is, in addition to this, inside the spleen, you're going to have a whole bunch of lymphocytes or white blood cells. Inside the liver, in all of these spaces, once you started filling them in, you would find hepatocytes. Because this is like the structural framework of a soft organ. They call that the stroma of a soft organ. That okay? Okay, so that's where you would find this tissue. Okay, and then we are ready to move on to the dense tissues. And we've got 3 dents as well. So we've got dense regular, dense irregular, and elastic. And I call it elastic dense. You don't have to put the dense if you don't want to, but it reminds me that it's both elastic and dense. Go ahead.
For reticular connective tissue, would writing framework as well as immunity?
It actually, you can write immunity if you want to, but it's not actually. It's the white blood cells within it would be doing the immunity,
Okay.
not the actual framework. Yeah, but I mean, I wouldn't count wrong. But you'll learn that deeply this semester as we start talking about this stuff. Okay, so let's look at the dense tissues and see if they all look dense first, right? So the dense tissues. So you've seen that slide before. How about this one? Does this one look dense to you?
Yeah.
As in more fibers than ground substance. How about this one? Does that look dense to you?
Yes.
Okay. How about this one?
Yeah.
Yeah, right? So it's kind of almost completely packed compared to, again, remember that we're comparing it against this, right? So those are the 3 dense tissues. And the dense tissues have lots more fibers than loose tissues. So let's talk about the dense regular. So if you look at this slide, we can see that it's dense. What about this slide makes it regular? Yeah, all the fibers are parallel to one another, right? That makes it regular. So look at this slide and tell me on this slide, is this tissue going to be strong from multiple directions or primarily this direction, right? Right? Just this direction, right? It's not going to be strong if you pull it from this direction. So the metaphor here would be like taking all of those collagen rope fibers and weaving them together so that you have a really strong rope, right? Okay, so it's strong from these two directions. So let's identify a few things in here. What is the cell called that's smashed between the fibers? That's called a fibroblast, exactly. And then what are the fibers? Yeah, collagen fibers. So this tissue is strong, but it's not stretchy, okay? You find this tissue forming most ligaments and tendons in your body, okay? Anybody know the difference between a ligament and a tendon? Which, what does a tendon connect? Muscle to bone. What does a ligament connect? Bone to bone. Okay, so they're made of the same tissue, but they just connect different things. So I want to talk about why you would want a rope for a tendon and not a bungee cord. Okay, so why are they not stretchy? Okay, so I want you to imagine the tendon that connects my biceps brachii to my radius and ulna here. Okay, and so what they're supposed to do is when the muscle contracts, it's supposed to move, right? So why do I want a rope here instead of a bungee cord? So how quickly do I want it to respond once I stimulate this to contract? I'm going to stimulate this to contract, and if I had to contract this and also deal with the stretch, do you see that there would be a lag time between the stimulus and the actual response, which would make movement relatively unpredictable? Would there be any benefit? Well, yeah, you'd be less likely to tear the damn thing, right? They have a little more give to them when you hit them from a position that you didn't expect. Like if they were made of bungee cords, you wouldn't see as many anterior cruciate ligament tears, for instance, right? But they also wouldn't have as much responsiveness when you're actually causing movement. Does that make sense? So it's both good and bad. It's definitely good functionally. It's just not particularly helpful when you have damage. So this particular tissue is strong but not stretchy, and it's primarily strong in the orientation that the fibers are running and not in other orientations. So you find this tissue forming tendons, ligaments, and what we call an aponeurosis. Aponeurosis is just a sheet of tendon instead of a cable of tendon. So a couple of locations where you would have these. You have this muscle right here called the frontalis muscle. You have this muscle right here called the occipitalis muscle. And they're connected to one another. And I could put a whole bunch of cables between them, but instead I've just got a big sheet of tendon. That's what an aponeurosis is. You also have an aponeurosis over your abdominal muscles and like inserting onto one another. Does that make sense? So that's all it really is. Okay, so next one is dense irregular connective tissue. And dense irregular connective tissue is usually really vascular. It's kind of hard to see the blood vessels in it, but it's usually pretty vascular. And it has bundled collagen fibers, meaning these are just individual collagen fibers running a bunch of different directions. That doesn't make a whole lot of strength. When you look at dense irregular connective tissue, it has bundles. Do you see these bundles of collagen fibers running a bunch of different directions? Okay, so this tissue is going to be strong in multiple directions, right? Not just one. So the metaphor that works here is take those same rope fibers and make a hammock instead of making a single rope, right? So it's strong in a bunch of different directions. So when I told you to think about irregular, I said, okay, if you want to feel what irregular feels like, pull up on your skin. You can see that little give that you've got, a little snapback. But if you want to feel what this feels like, dense, irregular, push on your skin and try to stretch it. And underneath the epidermis that is moving a little bit, there is a relatively immobile dermis that's really not stretching when you do that, right? And that is made of dense, irregular connective tissue, okay? So the reason, gross as it seems, that we make leather out of animal skin is because we are using their dermis, that really strong in multiple different directions, dense, irregular connective tissue, all right? All we do is moisturize it so that it stays pliable, and then you can use that tissue for lots and lots of different things, right? Kind of gross, but true. And then we also have dense irregular connective tissue on the outsides of things that you're going to pull on. Like on the outside of my femur, I can pull on it from this direction, that direction, this direction, that direction. So what kind of tissue should I wrap it with so that the tendons can insert onto it? On the outside of the bone, the periosteum is made of dense irregular connective tissue. Does that make sense? Mm-hmm. That's what I actually pull on. Also, perichondrium around the outside of cartilage is made of that. So dense irregular connective tissue is not super consistent in the way that it looks because it's super duper irregular. This is my favorite slide. This is the slide that they have in your textbook. So all you really need to do is to be able to tell it from other things. It's not regular. It's definitely dense, right? Okay. It's not elastic. So you can do process of elimination. You can figure that one out. The only thing that you might confuse it with is cartilage, but once I teach you cartilage, you sure as hell won't again. And then this one, just process of elimination, right? Can you do it by process of elimination? Yeah. And then this is just gorgeous. It's just perfect. I love that slide. Okay. And then the last of the propers is elastic or elastic dense. I always call it elastic dense because that's the way that I learned it, and also it helps me remember what it should look like because it's both elastic and dense. This slide is the slide in your textbook, which I think just looks like trash. I don't like it because they label this and they're like, oh, that's a fibroblast nucleus. I'm like, that's a blob in the middle of a bunch of other damn blobs. I don't even believe you. I think you arbitrarily labeled that blob. But if you look at this slide right here, can you not see some nuclei of actual cells? Okay, so this one has what kind of fibers? Yeah, elastic fibers running parallel. Not as nice and neat as dense regular, right? Wiggly fibers kind of running parallel. To me, this looks like a French pastry, the ones with the cream in between, right? Do you know those pastries, right? It looks like, it makes me hungry. I never ate before this class. I'm always freaking starving before I was like, oh yeah. Somebody else was like, no, that looks like sushi wrapping. I'm like, sushi. So this, and then you have the cells visible between it, so elastic fibers. And this tissue is kind of like a bungee cord, yeah? So you take individual elastic fibers, but you weave them all relatively parallel, and then you get some collective strength, but you also have some snapback to it, right? So you find this tissue in locations in which you need strength and stretchiness together, okay? Hooking two things together. So yes, most ligaments are made of dense regular, but the ligaments between your vertebrae need strength and they need some elasticity and stretch to them. There's a lot of movements that are going to occur there. Those are called your intervertebral ligaments, yeah, between your vertebrae. You also find this tissue forming your vocal cords, yeah, and you also find this tissue, the elastic ligaments of the penis will make you understand it. So during an erection, in addition to, of course, the hydraulics that cause erections, what you're going to have is you're going to have some ligaments that are holding on to the penis. And they need to be strong, but if you're doing it right, they also should be stretchy, right? Because you should be wiggling a little bit. You should, right? I mean, I realize you can do it the other way, but ain't nobody going to write a book about you if you do. So the elastic ligaments in the penis are made of these, which is dense, elastic dense connective tissue. So true vocal cords, ligaments between the vertebrae, and the elastic ligaments of the penis. Very strong, very elastic fibers instead of collagen. Okay, and then, can you tell that this one is, even though it's not as good a slide as the other one, once you have looked at this one, and this one, and this one, do you see that is elastic dense connective tissue? So you need to, once you think you know one picture, you need to move on to a different iteration of that same histology slide, wherever you can find it, right? Because memorizing a single image does not give you the flexibility to find it in an unfamiliar image. Does that make sense to you? So yes, most people go into Open Lab and they only study that histology book, and that's silliness, right? I just would not do that. I would study the histology book and then I would find something online. There's iPads in lab. You can actually take them off the stands and use them, by the way. There's one that students can use all the time. You've also got your own sources for. Okay, so next we are going to move on to the supporting connective tissues. And the supporting connective tissues, these are squishy. These are mostly kind of solidish. and these are going to be fluid, or there's only going to be one of those. So I'm going to erase that. And with the supporting connective tissues, there is going to be cartilage and bone. There are 3 types of cartilage. We're going to do that first. Okay. There are 2 types of bone. You only learn one histology slide in this class right now, but in the next chapter you're going to learn the other one. So I'm just going to go ahead and tell you what it is. Okay. So the histology slide you're going to immediately be responsible for is compact bone. Same one as in lab. And we won't learn this one today, but we'll prep for learning it in the future, and that's just spongy bone. Okay? And then the 3 kinds of cartilage are hyaline, elastic, hyaline cartilage, elastic cartilage, and fibrocartilage. Good? Okay, so what I want to do is I want to make sure that you can recognize any cartilage before you can recognize specific cartilages, because there is something really characteristic about cartilage that comes from the way that it actually develops. And that is, let's just look at this picture while we're talking, and specifically you're looking at this region of this picture. So the way that cartilage develops gives it one of its characteristics that's true for all 3 kinds of cartilage. So here's how cartilage develops. I have a cell in an immature cartilage. I'm going to call this cell a chondroblast while it's still maturing. Yes? Okay. What this chondroblast is going to do is it's going to make collagen fibers and or elastic fibers and secrete them from the cell around itself, right? And then it's going to pull in sugars and proteins from the bloodstream, and it's going to dump them on top of those proteins. So what you get is you get this gel that forms, yeah? And the gel that forms is relatively solid. It's got a little bend to it. It's kind of like Jell-O, because as gross as it is, Jell-O is made from the boiled down connective tissue of primarily mammals. Did you know that? I think somebody should have told me before I ate all that damn Jell-O as a kid, because I would have opted out. That's some grody ass stuff right there. So yes, you boil down the connective tissues to get all those collagen fibers, you reconstitute them, and then they actually make Jell-O. So what happens in this situation is you've got the Jell-O making itself. Okay. So what happens is, I want you to think about Jell-O salad, because we're in Utah, right? I thought there was going to be Jell-O salad at every damn potluck that I went to in Utah, and I have yet to see a Jell-O salad. Gotta tell you, what? I know they're super grody. So, do you see them at your potlucks? You must have old people at your potlucks. You got some old people? Okay, all right. So, I want you to imagine Jell-O salad. This little bastard's gonna have grapes in it. I don't know what the hell you were thinking. It's a bad idea. Everybody disapproves. Okay, grape. Grape is the cell, but the grape is going to form the matrix around it, right? So what it's going to do is it's going to make the matrix around it. It's going to make the Jell-O around it, right? But it has to leave itself a little grape home. And the little grape home, yes, is where it will sit. Pull it out, stick it back in there, still grape house in there, right? That's the lacuna, yeah? Okay, so when you make the matrix, you always leave a place for yourself to sit, right? The lacuna always surrounds them. And how that manifests when you look at histology slides is that the cells in cartilage almost invariably look like they have a frame around them. Yes?
I just wanted to share a little mnemonic.
Yes,
I
go
had
ahead.
a lot of trouble with that.
Yeah.
Someone in my last class, or my lab class, called it a kakuna. So lacuna, kakuna.
Lacuna, kakuna.
That's
Lacuna,
making sense. So-
lacuna means little lake. Okay, it's actually not a lake. Lacuna surrounds it. So, yeah, like a cocoon. Every cell has a little cocoon surrounding it. So they got kind of like a frame around them. So I want you to look and see if you can see that in these different slides. Regardless of the type of cartilage, cartilage is consistent about the cells having a frame around it. Now, I'm not saying the frame is always empty, right? If you don't get any fluid surrounding it, then it's going to be bright white around it. But it's okay if you get a little fluid in there, right? It's still got, what about this one? Can you see the frame surrounding the cells? Here, of course you can. And then this is a different type of cartilage, but it still has that, doesn't it? The cells are all framed, right? And you don't see that in other ones. This is a little harder to see, but do you see the frames around these cells? Yeah? And then last but not least, this one. Do you see the cells with little bright frames around them, right? And this is the best example, right? Cells with these bright frames. So what you want to be able to do before you worry about which cartilage it is can you tell it's a cartilage? Because of course that narrows it down to 3, right? So hopefully you can tell that it's a cartilage now. And now all we have to do is tell the three cartilages from one another. Yeah, go ahead, Anthony.
So the chondrocyte is that cell within the lacuna?
Yes.
Okay.
So it's the grape, right? The hole that the grape sits in is actually the lacuna, right? Good. And then all of this is matrix, and we can figure out what's actually in the matrix for just a second. Okay, so surrounding the chondrocytes and the lacuna is going to be the cartilage matrix. And the cartilage matrix, just to give you a visual, is primarily protein fibers. Even if you can't label them, they have to have fibers in them because that's actually how you make a gel. Okay, so don't worry that you can't label them in here. Protein fibers with solidifying, it's mostly sugars and protein, they're called proteoglycans, but you don't care. So around this is going to be the matrix. Now, sometimes the matrix is nice and smooth and you really don't see any labelable fibers. That is hyaline cartilage. Hyaline means glassy, as in the matrix is really smooth, okay? Hyaline cartilage. This is also hyaline cartilage, okay? And this is hyaline cartilage. This is the perichondrium that feeds it, but this is the hyaline cartilage, okay, over here. So hyaline cartilage has no visible matrix, visible fibers in the matrix. Now contrast that with this, that definitely has visible fibers, or this, that definitely has visible fibers. Still a cartilage, but visible fibers in the matrix. Good? Okay, so hyaline cartilage and all the cartilages have really, really poor blood flow, and this ends up mattering clinically. So hyaline cartilage, all cartilages are thicker than microscopic, right? Otherwise they wouldn't do a damn bit of good, right? So they're relatively thick tissues, right? You've run into cartilage, right? You've seen it or you've accidentally eaten it when you were eating meat, which super grosses you out and makes you want to be a vegetarian if you're me. So every single time, every time I teach this lecture, I'm like, this is so gross. Definitely going to do it this time. My kid's a vegetarian, so I have to make vegetarian meals every single time. I just can't do it because I'm actually a carbitarian. I don't even like vegetables that much. And if you just eat carbs, you get ginormous, right? So I'm too vain to be ethical, is what I'm running into, right? That's it, right? Damn it. I wish I were a different human, but this is all I got. The cartilage, when you get it in your mouth, it's kind of springy. It doesn't really give. Can't really break it down. But I want you to understand it has really poor blood flow because really the only place that you have good blood flow to any of the cartilages is around the outside of the cartilage in the perichondrium. So if you damage a cartilage, like piercing your ear or something, right, or just damaging your AC or damaging your meniscus, you're not going to get good healing because the tissue is too thick for diffusion. So what will happen is you'll really only get good healing at the edges that have good blood supply, and the middle is just going to be damaged forever, right? Anybody in here damaged cartilage and still have damaged cartilage? Yeah? Does it really ever get back to its original position? Go ahead.
Is the perichondrium made up of dense, irregular?
Yes, dense irregular connective tissue on the outside. And so as far as it can diffuse in, it diffuses in, and you might get some regeneration, and then in the middle, right, it really doesn't get appreciable amounts of blood flow. So we can use that to our advantage, and then sometimes it's a disadvantage. This is, again, hyaline cartilage. Hyaline cartilage is found at the ends of almost all of your bones, where the bones articulate with other bones, right? So 2 bones, or 3 here. So if I'm forming the glenohumeral joint,
Sorry.
oh, I thought that was mine.
Siri.
Yeah, Siri, he talks to me all the time. He just misses me while I'm teaching, I'm pretty sure. He's Australian. I'm fairly certain he's blonde, over 6'5". See? You missed me. What? What'd you say? This tissue, so if you're forming your glenohumeral joint right here, you're going to have cartilage at the end of the humerus, yes? You're also going to have cartilage at the end of the clavicle and cartilage at the end of the scapula. Because you don't want bones pushing against bones because you develop bone spurs when that occurs. So you always have cartilage sort of at the intermediary between those. So you have it also, if you want to feel what hyaline cartilage feels like, the tip of your nose right here. So this bone, obviously. But this is hyaline cartilage. Got more give to it than bone, right? But less than this is elastic cartilage. Spring, 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 not the bottom. That's just basically dermis and epidermis. Right? This is elastic cartilage.
There's no cartilage in the bottom of your ear.
Nope. Can you feel the difference? Just go up until you start feeling cartilage in the middle. It's right about here. Right? Yeah. So this one can heal all the way and close all the way. You won't be able to feel it later. And then during fetal development, your fetal skeleton, a cartilage model is made before it actually is bone. And then throughout the process of fetal development, and even after you're born, you are slowly but steadily replacing the cartilage with bone. And it will be mostly ossified by the time you're about 2, with the exception of growth plates, which are still cartilaginous until you're about 25. So 22 to 25 about. Does that make sense? So the entire fetal skeleton is cartilaginous, almost the entire fetal skeleton is cartilaginous first. Now, of course, it sucks when cartilage doesn't heal when you damage it on accident, but sometimes we use it to our advantage. Like rhinoplasty, when people have a nose job, I'm using the fact that I know that cartilage won't heal, right? Pulling the skin back, shaving down the cartilage and reshaping it, putting the skin back in position. If cartilage was regenerative, that would have been a waste of thousands of dollars. Correct? Right? But it also means, since cartilage isn't regenerative, how long does a bad nose job last? Forever. Exactly. Right? And then you can go in and have another one, but all they can do is take more off. And then you end up like Michael Jackson. Right? Who just keeps losing the cartilage tissue in his nose. Or my husband's ex-wife. But I'm pretty sure that was just karma. Because she's horrible. So she had 3 nose jobs, and now it's like so far up that I'm pretty sure she will drown when it rains. So I'm just waiting. Serves every bit of it. Okay, so see, I'm not a good person. I'm just me. That's all I got. Not a vegetarian, also kind of vindictive. There you have it. Going to be a vegetarian in the future, still be vindictive. Okay, so that's hyaline cartilage. Elastic cartilage is the next one, and this is not that one. This is the Springy cartilage. And elastic cartilage is found in the epiglottis, which keeps you from literally inhaling your food, right? It's also found in the inner ear tubes called the eustachian tubes. I don't know why the hell those need to be elastic, but that's what it's made of, right? And it's also found in the pinna of the ear, this external portion of the ear that's called the pinna, okay? And so you can tell that it's cartilage. What is this cell called? Chondrocyte. What's the space around it? Lacuna. What are these fibers? Elastic fibers. And can you tell it even if it's not that picture? Yes, right? Even if the fibers are a little denser than you expected them to be, it's not a dense tissue because I know what the 3 dense tissues look like, and none of them have frames around their cells. Does that make sense? And then this one, of course, is elastic cartilage. Okay, a really common mistake that people make, but they actually know the answer. When you put elastic, make sure that you specify whether you're talking about elastic connective tissue or elastic cartilage. Elastic alone will get the whole damn thing wrong because I don't know if you knew what you were talking about, right? So elastic cartilage or elastic connective tissue, of course, they both have elastic fibers in them, right? So you have to, they both are elastic. So that's elastic cartilage. And then the last one, which is the strongest one, has collagen fibers running parallel in it, and this is fibrocartilage. And fibrocartilage, still chondrocyte, still lacuna, all collagen fibers that you can see. Usually, for whatever the hell reason, stains blue like this. There are a couple times I've seen it stained other colors, so don't let blue be your only indicator. You still want the cells with frames around them and then parallel fibers, and that's what tells me that it's fibrocartilage. Then this is a little further away. You can see that one. So that's fibrocartilage. The only one that you might confuse this with is probably elastic dents, but please, again, note that the cells do not have frames around them. No lacunae in elastic dents, right? So I know that you know this, but I'm going to reiterate that whenever 2 tissues look hard to distinguish, stick them back to back as many times as it takes until they get easy to distinguish, right? So where do you find fibrocartilage? You find it between 2 bones that have an extraordinary amount of pressure between them. So your intervertebral discs between your vertebrae are made of fibrocartilage. Your pubic symphysis, as we developed bipedalism, there's a lot more pressure right here. That's made of fibrocartilage, right? The cartilage right here between your maxilla and your mandible is made of fibrocartilage. And then again, as bipedalism developed, our knees take an inordinate amount of stress, right? Because most of your body weight is actually up here. So what happened in our knees is you can see it develop with bipedalism. In addition to the hyaline cartilage that's at the end of the bones, the femur and the tibia and the fibula, We also developed 2 lateral pads called the lateral and the medial meniscus, right? Okay, they're lateral to the leg, but one's lateral on the leg and the other one's medial on the leg. And those are fibrocartilage pads. So that's in addition to all of the regular joint stuff. The knee developed more, right? More cartilage. So that's fibrocartilage, and it's the strongest of the cartilages that you're going to run into. Now, I see this happen a lot. Hopefully you guys won't do this. If I put name a body location for fibrocartilage and you put vertebra, are you going to get it right?
No.
Because what did you mean to say? You can say intervertebral disc. You can even say between the vertebra. That's fine, but definitely not the vertebra itself, which is made of what tissue? Bone, exactly, right? So you have to say what you mean, right? And even if I know that you meant something different, you don't get credit for it unless you actually say something different, okay? All right, and then the last of the supporting connective tissues is bone, also called osseous tissue, and this is the figure in your textbook. Okay, go ahead. Mm-hmm.
Why is why does it not have perichondrium
Oh, it does. You just didn't see it in the picture.
on the fibrocartilage?
Yeah, fibrocartilage. Uh-huh.
It does have it?
Perichondrium around the outside of fibrocartilage? Yes, it does.
Oh. Okay.
Why? Does it say that it doesn't?
I was distinguishing that there were in lab, there was 2 of the, which one didn't have perichondrium?
They've all 3 got perichondrium around the outside. Mm-hmm.
Yeah, I
Yeah.
thought it was.
It looks different in the 3 of them, but you don't usually see it in the other 2, right? They usually don't just get histology slides of it with it.
Yeah, in the lab, it says there's no perichondrium on the fibrocartilage.
That's not true.
Not true?
Mm-mm. It's not. But I've never actually made a histology slide with it visible. So maybe they mean there's no visible perichondrium.
Okay, thank you.
Yeah, that would be true. It's really hard to get the actual fibrocartilage to stain and the perichondrium around the outside to stain. Okay, so osseous tissue or bone. Okay, there's actually 2 different kinds. You're only responsible for 1 histology slide now. And again, the one that you had in your textbook, it looks like this or this. I think this is a really pretty slide. That one's kind of nice too. But before I actually show you what this slide and all the characteristics, I want you to understand how this picture corresponds to this whole bone, right? So where is that? What the hell are you actually looking at? How did I get that slide out of that bone? So the thing that I want you to understand is first the difference just conceptually between compact bone and spongy bone. First, what you are going to do with bone is the chunky part of bone is actually made of these columns, okay? And these columns are called osteons, okay? And so what you look at in a cross-section looks like a circle, but what you're actually looking at is a cross-section of column. So think of it like a tree. You know how a tree grows in rings, right? Same metaphor that you've got here. So what's the difference between compact and spongy? Well, if I have compact bone, what I have is all the trees are smashed right next to one another, hence compact bone, okay? And so it's going to be really dense and really strong, but also really heavy, right? Whereas as if it's spongy bone, they're running at angles to one another. And you get a bunch of spaces in between. They're usually filled with marrow, right? And it's less heavy. It's also not quite as strong. Still pretty strong, right? Does that make sense? That's the difference between compact bone and spongy bone. So now what I want you to understand is I want you to think of a long bone like, well, that one's the humerus, okay? Think about the humerus. Let's mentally do a cross-section of the humerus and look at the gross anatomy before we look at the microscopic anatomy. Ready? So if we do a cross-section of the humerus, it would look basically, right? Like I've got this on the outside. This is going to be where the compact bone is, right? From here to here. What's in the middle? Yeah, that's a marrow cavity, exactly. Okay. Okay. But that is not what you're looking at with the histology slide, even though this is round with concentric circles as well. What you're looking at the histology slide is, if I looked closely enough, I would see that this compact bone is made of a whole bunch of little crowded circles, right? Each with a dark spot in the middle. What I want to do is I want to do a histology slide of one of those. Yeah? So I'm going to blow that up, and I'm going to come up with that slide, right? And it's going to have big round circle, dark thing in the middle. Only this dark thing in the middle is not marrow cavity. What is it? Yeah, it's the central canal. What is in there? Not marrow. What is in there is blood vessels and nerves. That's how you are going to feed this entire tree. Yes. So this is collectively, this whole thing right here is called the osteon. Yes, that's the osteon, right? So how many marrow cavities are there in a humerus? Marrow cavities. One. How many osteons are there in the humerus?
Several.
Tons, right? Does that make sense? So that's what you're looking at when you look at this histology slide, which is important because we're about to do bone and we're going to do it in more detail and you have to know more about it. And understanding the structure helps you get the big picture. But of course, for lab, what you primarily had to do is go, oh, that slide, I recognize it. It's compact bone. But you didn't have any idea what the hell it had to do with anything. So the central canal in the middle is where the blood vessels and nerves run, and they are going to give nutrients to this entire osteon tree. And it's going to grow in rings, kind of like a tree. I'm going to have one ring, another ring, and another ring. Okay? Let's talk about the rings, both what to label and also what is functionally true about them. Anybody know what one layer of matrix is called in an osteon? Lamella. Yeah, lamella is singular, lamellae is plural. Okay, so I want to tell you what the matrix is made of so that you actually understand why bone is relatively solid. So what I'm going to have, start the same kind of story that I did in the cartilage, is I'm going to have a cell, okay? The cell is immature first, let's call it an osteoblast. And what it's going to do is it's going to start to make matrix around it, yes? It's going to make collagen fibers around it, okay? And then it's going to pull from your bloodstream, from your diet, calcium ion compounds, like calcium carbonate and calcium phosphate. It's going to use those and it's going to dump them onto the collagen fibers. So the metaphor that I want you to think about for bone matrix is reinforced concrete. How do you make reinforced concrete? What goes down before the concrete? Rebar, right? The rebar, the steel bars, right? And then you pour on the concrete and then it solidifies. What's the benefit of reinforced concrete versus unreinforced concrete? It's stronger, it's less brittle, it's less likely to crack, it's got a little more give to it before it breaks. Same thing with bone. So with the metaphor, the collagen is the rebar, yeah? Put that down first. And the calcium crystals that will solidify are the concrete, right? And then you get you get flexibility a little bit, like bone bends before it breaks, right? And you also get strength. So each of these is a lamella of bone matrix, right? And then once the cell is completely surrounded, it leaves itself a little pocket. What's the little pocket called? For the cell to sit in? Lacuna, right? So this is an osteocyte once it's completely surrounded and it's sitting in a lacuna. Yeah? Okay. Now, the lacuna are not usually very easy to see here, but conceptually there has to be a place in that solid for the non-solid cell to sit. Yes? So it has to have a place. It leaves itself a place when it makes all of that. So what you're going to see is you're going to see the way that this bone grows because I've got a whole bunch of osteocytes in lacuna all the way around until you get bored drawing, right? And then one last thing that I have to take care of, diffusion will not occur very readily from here to here because this is solid. So I have to make little canals through the solid tissue for diffusion of nutrients and waste products. And what are these called? Canaliculi, which means little canal. Good? Does that make sense? Okay, so this slide is important to understand because, of course, you have it both in lecture and in lab. And then this slide is also going to be fundamental for the next set of notes, so I'll hold you responsible for that one as well. So master it quickly and get it out of the way, and then you will have done it. Cool. And of course, there's a million different questions you could ask about that slide. Go ahead.
So that was the compact bone?
That's compact bone.
Okay.
Okay. So the way, I mean, this is oversimplifying, but what spongy bone would look like is instead of a bunch of these things crowded together, they would be running at different angles. And in order to get something that looked like that, you would either have to section that one that way or that one that way. And there's room for marrow between them.
So where specifically is the spongy bone?
Spongy bone is a bunch of different places. It is going to be, do you see spongy bone right here? Right there? A bunch of different struts running a bunch of different ways. It is, so you always find compact bone around the outside because think about how good this bone would be to attach a ligament or a tendon to, right? It wouldn't be able to handle that kind of stress, right? So compact bone is around the outsides of all your bones. If you have a region of the bone that's going to undergo a lot of stress, then it's probably mostly compact bone, right? If you have regions that you don't undergo a lot of stress, you're going to have spongy bone in the middle of them, right? And so the spongy bone, the advantage to spongy bone is it's lighter, but it's not as strong. The advantage to compact bone is that it's stronger, but it's heavy. So you need to have the sweet spot between the two of them. Otherwise, your skeleton would be so damn heavy that the amount of musculature you'd have to move it would limit joint movement. So we had to hit the sweet spot between them. And then when you start losing your bone mass, and you actually do, so we think of our skeletons as kind of being done when we get done growing, because of course, you know, you grow from fetal development all the way up until you stop growing in height, which is 22 to 25. Sometimes in girls it's as early as 18, growing in height. But that doesn't necessarily mean your bones are done growing. First of all, they still grow after that. And if you think about that, so I have a 19-year-old son. He is 6'7". Yeah, Gavin is ginormous. But he's still like all spindly. I think he's done growing in height. But what's he going to look like when he's 25? Like his bones are going to thicken out a little bit, right?
Wow.
His jaw is going to fill out. His zygomatic arches are going to fill out. His bones are going to fill out. Of course, the rest of his body is going to fill out a little bit too. So there's bone thickening that still occurs even after they don't lengthen anymore. And then the other thing is your bones are really active. We don't think of them as being really active because you really never notice any change in them. But your bones are constantly undergoing remodeling because when you change your activity, your skeletal need has different structural needs. And then also, your skeleton is a bank for calcium and phosphorus. If I ever have inadequate amounts of calcium and phosphorus, I will just remove it from my skeleton to deliver it to my cardiac muscle or my neurons or wherever it needs to go. So you can lose skeletal mass, you can gain skeletal mass. Throughout all of your teens, 20s, and 30s, and probably even into your 40s, you won't notice any change in the mass of your skeleton, but you will eventually start to lose more skeletal mass than you replenish, especially if you're inactive, right? And eventually that can actually compromise function. You can get osteopenia and then osteoporosis. So even though it looks done and we don't think about it very much in adulthood, it is never really done. It will change based on how you use it. Okay, so that is osseous tissue or bone, and it's always working. We'll talk about quite a bit about it in the next chapter. Okay, and then the last of the connective tissues is easy at this level. It's actually not that easy, but it's fine at this level. We don't learn any detail about it really. It's just blood. So blood is a connective tissue because it comes from mesenchyme. It doesn't have fibers visible in the matrix or anything like that. It's primarily, the matrix is primarily plasma, which is around 90% water. But there are cells in it, so it's a tissue, right? And it's made from mesenchyme, so it's connective tissue. And so when you look at blood, really at this level, all you have to be able to do is to tell the three major cells and cell fragments and be able to identify the plasma. So the plasma is wherever you don't see cells, right? It's not empty, it's actually fluid, okay? And then we've got three categories of cells that we run into. We have the red cells that are abundant and really, really regular in size, and generally red because of the hemoglobin. And they're easy to identify, right? And you can't call them red blood cells anymore. You have to call them erythrocytes, which means red cell, okay? Erythrocytes. Their primary function is carrying gas around, okay? Then the white blood cells There are 5 different categories. That's why they don't all look the same, but you can recognize them by the fact that they always have nuclei. And they're bigger than the red blood cells and less abundant. So even though the nuclei change in shape, they're still visible nuclei. Those are called leukocytes. And eventually you will label the leukocytes individually. But we'll do that in the cardiovascular system when you have a little bit more time and fewer tissues to look at. So those are called leukocytes. Overriding functionality of leukocytes is immune system and defense. And super duper complicated, but really interesting. Oh, there is a Netflix show. Oh my gosh. I can't even believe it's so good. There is a Netflix show. It's an anime. It's a Japanese anime. It's overdubbed in English. And it is all about the immune system. And all of the bad guys are like different kinds of beasts, and then the individual characters are like, but they're specific. What? No, it's called Cells at Work.
I actually started watching that.
Oh my gosh, it was so good! It's so
really good.
specific.
It's really good.
I've been watching it with my son, and he's learned so much immunity from it. Because, for instance, you'll learn later, but like a basophil is a type of cell that's like specific to allergic responses, and then like a leukocyte is general, and then you have your neutrophils that do one thing and like attack bacteria, but they can't do anything about viruses. So good, and the immune system is so hard. So like I would totally just watch that as I was taking micro. I'd be like, what the hell's going on? I think I have to go watch a cartoon about it because I don't understand what the hell you're talking about. So go watch that cartoon. It's really good. So my 13-year-old learned so much about the immune system. He's like, Mom, are basophils only involved in allergic responses? I'm like, and parasites? He's like, and parasites, yes! Like, hell yeah. Okay, so later on you'll get that. But leukocytes. And then leuko means white. And then the last ones are the platelets, which we won't call platelets anymore. We'll call thrombocytes. And thrombo means clot. Okay? So these are your clot cells. But they're actually not whole cells. They're the little teeny things, dark staining. They're actually fragments of cells. They used to be a cell, and then they busted apart. And their primary function is to initiate blood clotting. Blood clotting is this really long, complicated cascade. But platelets get it started, okay? So these are thrombocytes or platelets. You don't have any trouble recognizing them, right? So it's a really, really easy slide, at least right now. Later on, when you have to recognize the individual type of white blood cells, it gets a little harder, but it's not that hard even then. So those are thrombocytes. Okay, so last little thing that we're going to do, and then we'll move into nervous tissue, is the same. Go ahead. What about lymph? Lymph is not a tissue. So we don't study it as a tissue because the cells that are in lymph actually come from blood.
Okay.
So they're made by the bone marrow, made by the blood, and then they migrate into the lymphatic tissue. So primarily what lymph, that's actually a really good question. So what lymph is primarily just extracellular fluid, right, that white blood cells move through on their way from one to the other. It doesn't like replenish itself, and it doesn't function individually as a tissue. It's like a highway, right? But exclusively for lymphocytes and the antigens that they produce. They haven't touched on the lymphatic system in that, have they? Did you watch the whole thing? I've only watched like the first 4.
I just got that part of the
You
colon
did?
disease.
I'm
you know,
so
colon-
excited.
so-
Yeah, and my son was just watching it with me, and my husband just walks by and like rolls his eyes because like I'm cooking dinner yesterday and I'm listening to a podcast about infectious diseases, and my son is asking me about like this infectious disease, and then later on we were watching this, and he's like, "You two are so freaking weird." I'm like, I needed another one of me in the world, so I made one. It's the best thing ever. Only he's funnier. He's so much funnier than I am. I needed a companion. Of course, he's 13, so he's almost done with all of my shenanigans. So be like, you're not funny. And he started to tell me to chill. Oh my God. I'm like, I am going to strangle you. And he's like, chill, Mom. And I'm like, whoosh, throw something at him. Just so you know, that does not make anybody chill. Okay. Okay. So how do you know when you're done studying a connective tissue? Same thing that we did with epithelial tissues. These are all of the things that I can think that I could possibly ask you about a connective tissue. So if you look at any connective tissue, the first thing that you need to be able to do is, of course, figure out where it fits into that category, right? So which general type of tissue is it? Is it supporting? Is it fluid? Is it proper? What is this one? That's fluid, but what is this one? And what is this one? And what is this one? Proper. That's proper, right? So if just a quick way to remember, fluid, right? Supporting solids, right? And the proper is the squishy ones, right? Everything that's neither fluid nor solid is the proper. So it's kind of the catch-all for anything. And then, of course, if it is proper, loose versus dense. So what's this one? That one's dense. So you have to be able to do that. And you don't usually have to do those two things in lab. But again, lecture is really about holding the organization of things so that you can have recall later. So you always have to do more organization of the material in lecture than you do in lab. And it tends to make it more easily accessible later because you'll remember, oh, there were 3. What's the one that I forgot? And then you pull it up. At least that's the way my brain works. The specific tissue, so what most students stop at is name this tissue. But you are a good student, so you will not do that because you know that there are other questions that could be asked. But most students will name this tissue, and what is it? Elastic or elastic dense, or name this tissue here, hyaline cartilage, and then they'll be done, and they won't realize that there's a lot of other things that I could ask you. For instance, I could ask you, what is this structure? Lacuna. What is, of course, I could ask you that. I could also ask you, if we go all the way back to this one, I could ask you, what is this structure? Lipid vacuole, right? Anything that is labelable in it, you can actually be asked, okay? And then the other thing that students really, I don't know why, but students have such a hard time with name the cell and connective tissue. And I don't want that to be you, so I want you to know that that tends to be a weakness in students. So when you are looking at a connective tissue, you have to get into the habit of naming the cell. If you can't remember, what do you guess? Fibroblast. Because there's usually a fibroblast in there somewhere. You can pretend like you freaking knew that. You're like, oh, I thought, well, that tissue has fiber, so it must have fibroblasts, you know. So you can guess that. But, of course, sometimes you're wrong. Because what is this cell called? Adipocyte. What is this cell called? Fibroblast. That's a fibroblast. What is this cell called? Chondrocyte, right? And what is this cell called? Osteocyte. It's an osteocyte once you don't see any osteoblasts in here because this is done. This is done bone. Good? That makes sense? So make sure you can name the cell, and then you can move on with that one. And then fibers, and if there's visible matrix, you can label the matrix region, even the ground substance if you wanted to. I don't usually make people differentiate between matrix and ground substance because it's really hard to phrase that question. Like if you point to it, you're like, they're going to say matrix, and I would have to say, okay. So how do I write it? The stuff in here that is not cells and not fibers and is in that space, right? I'm like, well, I'll just like take both answers. Go ahead. Her and then you.
When you ask about labeling an osteocyte, we have to put osteocyte in the
No, if I say label the cell, you put osteocyte. If I say label the space surrounding that cell, you put lacuna. So in anatomy, it's like really, really a responsibility on me to phrase the question carefully enough that I get the answer, right? Okay, so if possible, you might have to be able to, of course, name the fibers. So what are these fibers? Collagen. And you can't really label the matrix there, but you can here, right? That's matrix or ground substance. Either one I'd have to take. And then, of course, the things that students hate is specific body locations. So let's do a little practice on specific body locations so that you guys will get a little more confident at it. So let's do a specific body location for this tissue. It is in the subcutaneous layer, also called fascia, right? So that's good. What about a specific body location for this one? Subcutaneous layer would work as well, right? Somebody put surrounding some organs. What about the person who put stomach?
Not specific enough.
Well, first of all, the stomach is an organ, and it doesn't have any of this tissue in it. If they had put subcutaneous layer of the abdominal region, somebody else put butt. And I totally know what you're talking about, but you didn't get me to the right place in my school bus. And also talk about your own damn butt. So you have to get me to the right place. And then what about this one? Tendons and ligaments, almost all of them, right? And what about this one? Meniscus, intervertebral discs, and what about last one? This one. Don't break your penis. Elastic ligaments of the penis, right? You can put a bunch of different places, right? Vocal cords. Good? Okay. So make sure that when you are studying, you are not just flipping through pictures. You are making sure check, 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 and then move on. And if you can do this with multiple iterations of the same tissue, then you can move on, and then there are no trick questions. Because this is literally all the questions I could think of that I could possibly ask off of a connective tissue. So, good? Okay. So now we are ready to move on to nervous tissue, but hold on just a second because I need to look for a slide. So just talk amongst yourselves. You guys are like 15 minutes ahead of the other class anyway. I just need to...
I actually have a question for you.
Sure.
I was curious
I'll
if
try.
the serous membranes are all dense irregular.
Serous membranes are loose irregular with simple squamous on top of them,
Okay.
is what those are. Yeah.
That's like the sac that's around the organs.
I just needed to find another picture, so just talk amongst yourselves. I'll be back with you in a minute. Oh, there it is. Yeah, I just realized that. Okay, that's better. Okay, so nervous tissue is what we are going to do next, and nervous tissue in your textbook looks like this, but it doesn't actually look like that, and you're not going to see slides that look like that anymore. I don't know why they did it up this close and made the cells stain so well that you could really see them, because what it generally looks like is more like this. Okay, so I want to tell you how you can tell that it's nervous tissue so that you can sort of get confident with looking at this instead of that. So what you are actually seeing when you look at nervous tissue is the cells. There's two different types of cells, or it's actually a bunch of different, but two different categories of cells, and they're really different sizes, right? You've got a big old space filler and then a bunch of little bitty dots. Well, the big old space filler has this vague suggestion of kind of a starburst shape, right? So you can see it really well that it's starburst shaped there, but you can just see the suggestion that it's got things sticking off of it right there. And then in the middle, usually, if you get a good one, you don't always get a good one, but if you get a good one, like this one right here, you can see, at least in some of them, what we call the owl eye nucleus, in which the nucleolus stains really dark and the nucleus is light. Okay, that's kind of characteristic of neurons. Now, it's not always super, super obvious, but it's a secondary thing that you can look for to kind of confirm the tissue that you're looking. Do you see the owl eye nucleus in this one? Dark nucleolus with a bright one around that. Okay, so you're going to have these cells, and they're big, and then you're going to have a bunch of little sort of nondescript, primarily just nuclei around them, right? So that's part of how you can tell this from a connective tissue, because otherwise it might look like a connective tissue, right? Two distinctly different types of cells, right? One of them is really big and not that abundant, the other one's much smaller and relatively abundant. Does that make sense? So let's name these two. This one is the star of the show, gets all of the attention, called a neuron. And then these guys are either called neuroglial or just glial cells, which means glue. We'll talk about structure and function of each one of those. So histology slides of nervous tissue are more likely to look like this than this, but FYI, this is just one part of the nervous system. If you looked at the spinal cord, it would look slightly different than the cerebral cortex. The white matter within the brain, it would look very different. So this is just like a getting you started on nervous tissue kind of thing. What you are going to see in this tissue is sort of this characteristic with the two different types of cells. So let me tell you a little bit about the tissue, and then we'll go back and label some more things. So what does nervous tissue do for you? Well, super duper complicated functionality, but let's simplify it first. So if I put my hand on a hot stove, what I need is for three things to occur. I need information in, I need to process the information to figure out what needs to be done about it, and then I need information out to command the change, right? So that's what nervous system does for you. It does sensory, which is information in, taking the information in. It does processing, which primarily happens in your brain and your spinal cord. And then it commands the actual motor response, which is moving the arm, right? So sensation, integration, and then response is what the nervous system does for you. And of course, that's a really, really simple one, right? It's kind of a reflex, right? Okay. But it's going to be super complicated, right? Sensation, integration, and response is happening right now. The sensation that you are taking in, right, is the sound of my voice and whatever you're seeing on the screen. And the integration is making little connections between things that you learned in lab or things that you learned before or whatever. And then the response is I understand it, I get it, or I'm writing it down, or I'm making a connection. So the response is actually some kind of output, right? So it can be super duper complicated. So where is most of your nervous tissue? Most of your nervous tissue is sequestered. And that's really important because these guys right here, the neurons, these are the stars. They're the ones that are really doing all of the electrical activity and releasing neurotransmitters, for instance. And then this is its support staff. The support staff is the glial cells. And the neuron does all of the really, really important nervous system stuff, but it can't work without its support staff at all. It's kind of like if you took the Olsen twins out of LA and put them onto the Serengeti, and you said, live. They would say, how? I have 6 people that do this for me, right? So I don't know anything. Neurons are so specialized that they need this entire support staff. So they're really, really specialized. They're super delicate. And the other thing is that they're amitotic. What do I mean by that? They don't go through cell division. So if you lose them, they are gone. Yes? So back to the vast majority of your neurons are sequestered within the brain, the spinal cord, where they are encased by bone, wrapped by 3 different layers of protective membranes called meninges, and then bathed constantly by a nutrient broth called the cerebrospinal fluid. Because if you lose them, they are mostly gone. Now, they found a few recently that are regenerative, and we still don't understand that, but mostly they're non-regenerative. So we have to protect them, right? And they're super duper duper important. So what is it that's going, if most of them are in my brain and spinal cord, what is it that's going from here to here? Wiring. It's primarily just extensions. The actual neuron itself is protected in here, right? But I've got wiring going to and from whatever organs, right? Because if I cut off the arm, the body still lives because the body was actually in the spinal cord, for instance. Does that make sense? So that's the nervous tissue that you've actually got right there. Brain, spinal cord, nerves, sense organs. Go ahead.
So say you get like a fracture in your spine, are you losing some of those cells and they're not regenerating?
Are you talking about a spinal cord injury?
Yeah, like
Okay.
reverse fracture or something like that. where
Well,
you're
if you
not
just
being
damage the
used.
bone, it might not have touched the actual nervous tissue, right? Because the bone is encasing. Although, of course, the nervous tissue is really encased in there, so damaging your spine and high likelihood of damaging your spinal cord. If you just have a fracture, it doesn't mean you damaged any nervous tissue at all, right? If you damage nervous tissue, like for instance, you have spinal cord injury, like skiing or whatever the hell, right? Spinal cord injury. If you actually cut through the spinal cord, permanent damage. Non-regenerative tissue. With the little asterisk, permanent damage, that embryonic stem cell treatment has worked for some people in experimental situations if you can get them to therapy quickly enough. Still experimental. When George Bush II was president, though, he removed all of our federal funding for embryonic stem cell research here in the United States, because he was confused and thought that embryonic cells came from aborted fetuses. They don't. They come from in vitro fertilization. He was not a smart man, and I know this because I'm from Texas and I voted against him every single time I had the opportunity, and then you people voted him in. I told you.
None of us.
I didn't.
No, none of you. Right. There's a few of us. But so anyway, he removed all of that. So we don't really have any appreciable amount of embryonic stem cell therapy treatments going on in the United States. We do have a few. But go ahead.
Can you talk about what happens in ALS then?
I can when we get to the nervous system.
Okay,
It's pretty complicated though.
Yeah.
But yeah, we can talk about it. Make sure you send me an email right before we start the nervous system, and I'll include it as one of the clinical things in the nervous system. Good? Okay, so let's see, what the hell was I talking about? Neurons, neuroglia. Oh, damage, right? So what happens if you lose nervous tissue? If I have a stroke and that part dies, it's gone. That's what happens. What happens if I decide to play football because I haven't been reading the news and get 72 concussions and lose part of my cerebral cortex? What happens? It's gone, right? And it's gone in your 20s. And there's a whole lot of years in which you are not going to be athletic or cute. You might as well be smart. A whole lot of years ahead of you, right? Okay. So, guess whose child does not get to play football? It's okay because he's a musician anyway. He's like, I don't know why people would do that. That looks dumb. I don't even want to stand next to that person, much less run into him. Damage them and they're gone. Let's talk about the neurons and neuroglia just a little bit and talk about neuron anatomy, and then I'll let you guys go. So when you look at neuron anatomy, so if you have a neuron that you can see all of it, which you usually can't, but if you had one that was super duper well stained and you could see the whole damn thing, you can have something that looks like this, right? You got the starburst shaped cell, and a stylized drawing of that might look like this. So let me just tell you what you're actually seeing when you look at that. When you look at that, one neuron, starburst shaped with processes, but one of the processes is much longer than the other. I don't think you can tell in that histology slide which one is the axon, but they say you can, whatever. So this one is called the axon. Its job is to carry 250 miles an hour an electrical stimulus that way. These are called dendrites, and they carry information in. So if you, for instance, put these in series, right, I could have one communicating with another, right, and the information would be coming in, going down that way, and then connecting with that one, right? Axons and dendrites, right? So this guy right here is the cell body of the axon. It's where almost all the organelles are, also called the soma. Good. Nucleus, nucleolus, nucleus, right? Owli usually looking nucleus. And the axons and the dendrites. So basically, every time you look at a picture of a neuron, if it's stylized enough or if it's labeled enough, you can tell the direction of information flow. So if we look at this figure, we could label which direction information was flowing through it. Which direction would it go? Yeah, it would go that direction, right? And so the good thing about understanding anatomy is that if I start putting in a bunch of neurons, you can look at the pattern and go, oh, it's all going that direction, or it's going that direction, right? So the structure-function relationship. So the neurons have axons and dendrites. And then the neuroglial cells, all those really are is this sort of complicated support staff for the neuron. And they make up half of the brain volume, although each one is tiny. There's just so many more of them. Those are still regenerative, meaning that, you tell me, if I get a cancer in my brain, is it more likely to be a neuroma or a glioma? Yes, it's more likely to be a glioma because those are still regenerative. So I'm not saying neuromas don't occur because every damn thing that could occur occurs. So they do a whole bunch of stuff, and this is where you can start talking about ALS, but later we will. They make myelin, which is this coating that makes the impulse go really quickly down the axon. They do phagocytosis because the Olsen twins cannot protect themselves. They bring the coffee, nourish the neurons because they don't know how they like their coffee. They put gas in the car. They also are responsible for a few diseases that manifest as neuron diseases, but only because their support staff petered out on them, right? So we will talk about those as we move through. Okay, I will see you guys on Thursday. We just have muscle to do and a little tiny bit of organs after that, and then we'll start bone.